Hello everybody, thank you for attending our first webinar of 2020. Welcome to the Biophysis webinar entitled Deep or Not Too Deep, which will be about sequencing depth in the field of microbiome analysis. This webinar will be presented by our Scientific Operations Manager, Thomas Carton. Before entering the topic, please let us introduce Biophysis. Biophysis is part of Institut Mérieux, an independent family-owned French group established in 1897, following in the footsteps of Louis Pasteur. Institut Mérieux is contributing its experience in biology to improve medicine and public health across the globe. To fight against infectious disease and cancer, the Institute imagines and develops new approaches in the fields of diagnostics, immunotherapy, food safety, and nutrition. Its three bioindustrial companies, bioMérieux, Transgène, and Marine Sciences, working closely with its entities devoted to innovation such as ABL and Mérieux Développement, have contributed to major advances in medicine and public health. Biophysis is Marine Sciences' global research organization, serving innovation in food, nutrition, health, cosmetics, and consumer goods by offering complementary, differentiating, and innovative services, including preclinical and clinical assessments and microbiota-related research. Based on our own clinical investigation centers and our central laboratory, Biophysis offers a global and continuous solution which enables clients to substitute their product in order to maximize the market impact. Biophysis serves innovation in the three main regions with dedicated teams in the USA, Asia, and Europe. We offer services through three connected business units: clinical research to help you conduct your clinical trial, central labs for all biological analysis including microbiome, and research and development to answer all of your specific requests. Regarding the microbiome side, Biophysis is the only one-stop shop solution, providing services in all steps of clinical and analytical operations. We can help you on study design, sampling kits production and logistics, clinical operations, next-generation sequencing, data mining, and valorization of the results. Our team integrates all expertises: biology, bioinformatics, biostatistics, and in the last years we conducted several preclinical and clinical projects on different microbiome as gut, vaginal, oral, and skin. And now, I will leave the microphone to Dr. Carton, who will start his presentation.
Hello everyone and welcome to this webinar whose aim is to address an NGS issue in the field of microbiome analysis, too deep or not too deep. First of all, we have to go deep into metagenomics and microbiome. As you should know, we are not alone, and if you have a look at different sites of your body, you will find billions of microorganisms which compose different communities whose domain are oral flora, the pulmonary microflora, the gut microbiota, the skin microflora, and the vaginal microflora. One of the main problems we encounter to study these microorganisms is that they are mainly not cultivable. So they cannot be easily studied through classic microbiology, through Pasteurian methods. It's the reason why in the last years the omics methodologies family replaces microbiology to study these communities of microorganisms. The omics family contains a lot of members like metabolomics, proteomics, lipidomics, and so on. And amongst all of these methodologies, you have to note that currently the winner is metagenomics by next generation sequencing. This is a winner because sequencing equipment encountered the NGS revolution with a huge increase of sequencing capabilities. For example, the 13 years and $3 billion required to sequence the first human genome decreased to 3 days and less than $1,000 today. So even if the microbiota genome, which is named the microbiome, contains more than 100 times as many genes you can find in a human genome, we are now able to analyze it easily by using NGS. On the other side, because it becomes easy to obtain a massive amount of DNA sequences, we sometimes sequence more than needed and forget the parsimony concept. On the place NGS has within the study of microorganisms, we can distinguish mainly three applications. First, whole genome sequencing application, where we start from an isolated microorganism and that is classified as a genotyping method, since the aim is to compare several strains for their genomes. WGS is mainly used in epidemiology. Secondly, metabarcoding application, also named 16S sequencing or targeted metagenomics. That corresponds to an inventory of the microorganisms which compose the studied microbiota. The inventory is done by targeting in all genomes the same gene whose DNA sequence varies between microorganisms. For example, the 16S gene for bacteria. Metabarcoding helps us to answer the question, who are there? The third application is shotgun metagenomics. That corresponds to the sequencing of all the genomes of the microorganisms within a microbiota, giving us information on both taxonomical and functional sides. So not only who are there, but also what can they do. Currently, shotgun metagenomics is about to completely replace metabarcoding because of its higher resolution and of a decrease of its cost. A very important thing is that whatever the application, whole genome sequencing, metabarcoding, or shotgun, and whatever the NGS equipment you use, one NGS run only delivers a limited number of raw reads. A raw read is a fixed size portion of the DNA molecule that have been deciphered within the famous alternation of the four bases, AGCT. For WGS and shotgun, these portions are taken randomly all along the genomes, and for metabarcoding, all the readings target the portion you choose, for example, the V3, V4 regions of the 16S gene. You have here 4 examples coming from the main sequencing platforms with a read number from 4 to 25 million reads per run. Of course, it will depend of the platform and also of the reagent you use within the platform. So it means that the sequencing depth is a sampling effort. The sequencing depth is how many reads per sample. And because of the fixed number of reads per run, the sequencing depth is also related to the number of samples you decide or you have to analyze at once on an NGS run. For WGS, it can correspond to the number of reads you need to completely cover the genome of a strain. And this is related to the size of the genome of the strain. We usually express the sequencing depth by the times we cover the strain whole genome. For example, 10 times the genome. For metabarcoding, the number of reads you will need to describe the population or to detect bacteria of interest. For example, you will need at least 100 reads if you want to detect a bacteria which is present at a relative abundance of 1%. And for shotgun, finally, the number of reads required to detect a function of interest or to go up to the strain level resolution. So it means that with a very high sequencing depth, you start doing WGS within shotgun analysis. In the next part of the presentation, I will focus on shotgun metagenomics, taking into account that we can partially link the taxonomic side of the shotgun analysis and the metabarcoding. But before calculating how many samples you will put onto your sequencing run, so how many reads you will have per sample, and because it's not really so simple, you should be aware of what came from your sequencing platform. Because raw depth, so the number of reads directly out of the sequencing machine, is not usable depth. First of all, the environment of the studied microbiota will have an impact because whatever the way you use to extract the DNA of the microorganisms from the biological matrix, the final DNA solution will still contain DNA from the host, and this DNA will conduct to unusable reads for your microbiota study. And even if from a fecal sample you will only have near 2 or 3% of host DNA, this contamination can increase up to 80% of host DNA for some other matrices like vaginal swab. So in this latest case, it means that you will lose 80% of the raw reads and that you will keep only 20% of raw reads to study the microbiome. Secondly, a feature of all NGS equipments is that, yes, they have a big capability, but they also make a lot of sequencing errors. Sometimes at more than one error every 100 bases which is the Q20 level. Currently, the highest quality level is of Q30, one error every 1,000 bases. So even if the bioinformatic pipeline should be able to eliminate the corrupted reads, these errors will conduct also to lost reads from the initial raw data set. Note that even if I only talk about reads, there is read and read, especially because they can be of very different size, and so they can contain different information quantities, from several hundreds bases for Illumina and ion torrent sequencing to the long-read technologies of PacBio and Oxford Nanopore. It means that the raw read number shall always be also considered in relation to the read size. And it's the reason why sometimes we do not talk about raw reads number, but about bases number. For example, 5 millions of 200 bases reads corresponds to 1 gigabases. The wet part of the whole analytical process is not the only step where you can lose reads. Indeed, on the bioinformatic part, the dry part, it exists thousands of bioinformatic tools for trimming, filtering, merging, removing chimera, and so on. And depending if you want to be conservative or not, various amount of reads will be kept. The more you will be conservative, the more you will risk to keep poor quality reads. On the other side, if you are too stringent on your quality control, you will start losing reads that could be informative. So this is clearly a critical choice that again conducts to lose reads and to decrease the ratio between usable reads and raw reads. At the later step, if we consider a clean data set of reads, so that does not contain reads that come from host DNA or poor quality reads, you have to assign them. So you have to choose a reference database and an assignment algorithm in order to identify your reads. And again, depending on your choice, you will lose reads. In this example, you will see that depending on the bioinformatic method you use, you can obtain very different assignment levels, from 10 to 20% assignment. Of course, it's only an assignment level, and some other performance criteria have to be evaluated, for example, the confidence of the assignment. So finally, be aware that the raw reads number can be completely different from the final usable reads number, with sometimes less than 5% of the raw dataset as a usable dataset. Unfortunately, it's easier for marketing reasons to sell raw reads. But in all cases, the question remains, will you have enough usable reads for your project? On the other side, don't you have too many materials for your project? Are you sure that this number of reads is the most suitable in financial, in operational, and of course environmental terms? Are you sure it is the best fit-for-purpose sequencing depth? So it's the reason why we have to go out of depth, because sequencing depth is only a tool and couldn't be a name. The goal is not to collect reads, it is to have the right ones, so we have to understand why we need more reads in some projects than in others. If we go back to matrix, you have to know that from identical depth, the more you lose reads because of the host DNA contamination, the less you can detect low-level microorganisms. Because microorganisms are present at very various levels, and some of them can be part of subdominant flora. So if the host contamination is too high, you maybe not have enough usable reads to detect these low abundance microorganisms. So the sequencing depth has to be in line with what you want to see within the targeted microbiota. For example, the same raw sequencing depth corresponds for gut and vaginal samples to very different levels of detection. This is why, once again, raw depth is not an objective, but must be adapted as the tool that it is. In the same way, if we go back to the taxonomy, just take a moment to consider the relationship between the targeted feature and the sequencing depth. the less the relative abundance will be, the more you will need to increase the sequencing depth, as shown in these rarefaction curves. In the upper graph, for the upper taxonomical levels, the features are at middle abundances. So you do not need a high sequencing depth to discover all the features. And on the lower graph, for the lower taxonomical ranks, the features are at low abundances. So you will need more reads to access all the features. This is generally valid between the different taxonomic levels and also within a level where coexist taxa of very different abundances. But sometimes we do not need to identify all the microorganisms. We only want to describe the microbiome ecosystem. In that way, you can use the tools that have been developed in ecology, the diversity indices, like the Shannon Index, Simpson, Fisher Alpha, and so on, that do not require a high sequencing depth. Of course, you can observe some variations of the composition by increasing the number of reads, as you can see in the graph on the left, which shows the composition of two samples with two different depths and a factor 10 times between the two depths. But as you can see on the right graph, if you use diversity estimators like Shannon Index, you will need less than 100,000 usable reads to describe robustly a microbiota at the ecosystem level, or if you only need to observe the main microorganisms. As previously said, shotgun metagenomics is the new gold standard of NGS analysis. So if we go back to metabolism and to the functional inventory you access for the shotgun metagenomics, in Biofortis we also confirmed with more different approaches what had been previously described in scientific papers for the 500,000 reads level. At this level, you will reach 97% of the information, and that could be enough for your project. Note that it is an amount of usable reads and not of raw reads. Considering all reads yet that you will lost because of the matrix, of the sequencing error, and so on, it means, for example, that you will need several millions reads to study a gut microbiome. Again, this may be more about usable reads number than raw reads number. So talking about sequencing depth and determining the right depth level means how deep for your project, how deep for your need. In Biofortis, we think that from all explained previously and from what we saw in various projects, we can really distinguish 5 different levels. The first one is the very low depth level, which is only applicable when the targets are over-dominant into the whole microbiome. For example, in a clinical side for the detection of pathogens in acute infection step, where the pathogens could represent up to 80% of the whole microbiota, as in this example. Of course, in these diagnostic applications, sequencing depth and accuracy are of less importance than sequencing speed. The second level is the low depth level that we named the Explorer level in Biofortis, where you will have access to the main microorganisms as well as to the ecosystem structure, for example, in order to evaluate the impact of a product onto the microbiota equilibrium. By using all tools coming from ecology and based on alpha and beta diversity evaluation, you can have a very good overview of what happened to the microbiome ecosystem equilibrium through the 3 dimensions of biodiversity. The richness, that corresponds to the number of species, the equitability or evenness, which is the repartition of microorganisms within the species, and the disparity, that corresponds to the genetic difference between species, so to the whole genetic luggage of a microbiome. Note that I do not give a defined raw depth because, as explained, it will depend on the matrix. The third level is the medium depth level with several millions reads, which is named the advanced level in Biofortis, because according to the literature and to our own expertise, it represents finally the best balance at the taxonomic and functional levels. This advanced level was determined compared to the 97% threshold previously shown. It means that by using the advanced level, you will have 97% of the functional and taxonomical informations within your microbiome samples. This level can be useful for secondary or exploratory endpoints in a clinical study. The fourth level is the one of the high depth level that we named the expert level in Biofortis, where you will be able to differentiate at the strain level, for example, in a lineage project of Bifidobacterium bifidum between mother and infant microbiota. Note that, as previously explained, the required depth for the strain level also depends on the strain's relative abundance amongst all other microorganisms, and of course will depend again of the matrix. And finally, we can cite the level number 5, the very high depth level, where you can detect the point mutations inside the genome of the microorganisms. Because of the huge amount of produced data and of required calculation capability, this sequencing depth should be limited to very specific topics because it is clearly oversized for a lot of projects with all the consequences in terms of wasted time, money, and energy. So as a conclusion, I hope that now it's pretty clear that a microbiome study is not only a question of sequencing depth, a question of row reads number. Each microbiome project has its own specificity and its own requirements regarding the design, the matrix, the biological question you have, and maybe can we help? So thank you for your attention. Too deep or not too deep? That was the question, and I see there are others.
Thank you very much, Thomas Carton, for this excellent presentation. We hope you enjoyed the presentation, and it was very interesting and create a lot of interaction on the chat platform. Don't hesitate to write your question on the platform. Dr. Carton won't be able to answer to all those questions you have on the allocated time, but we will do by email under 1 or 2 weeks. So now it's time for you, Mr. Carton, to answer the first question.
So the first question, your presentation didn't address metatranscriptomic. Why? Yes, I didn't address metatranscriptomic because of a technical limitation which explains there is not a lot of transcriptomic publications onto microbiome. Indeed, when you extract RNA, you obtain a solution that contains mainly ribosomal RNAs, more than 90%, and a very low proportion of messenger RNAs. So you need to deplete the ribosomal RNAs to concentrate the messengers, because only messengers RNA are informative. And on the contrary to human messenger RNAs, the ones of microbiota do not have a poly A tail. So you need to use either an enzymatic solution or a capture system very less efficient and more complex and more costly than for human RNA. What is the advantage of PacBio and Oxford nanopores since they are the ones with the highest error rate? Yes, as you saw in a following slide after those on the error rate, PacBio and Oxford are also the ones with the longest reads. So it can be of interest. Moreover, the errors of PacBio and Oxford are more aleatory than in Illumina and ion torrent sequencing, which make errors in a specific way, for example, in the JC-rich sequences. So it means that by multiple alignment, you will be able to distinguish and correct these random errors, and finally reach Q30 after this bioinformatics step of correction. And of course, you will lose reads to correct the errors. So it's the reason why you cannot choose on only one performance criteria, but that you have to weigh strengths and weaknesses of all methodologies. So finally, what I understood is that if I can have more read than needed, it will be better for my project. Is that true? Of course, no. First of all, I can't be agree with this waste of resources. And secondly, you have to be careful not having too many data and not enough samples if you want to be able to conclude in a statistically significant manner. This is the well-known curse of dimensionality. And the last question, how did you determine your sequencing depths and why are they different between laboratories? Yes, they are different. As I try to explain, there is a lot of different steps that impact the ratio between raw depth and usable depth. In Biofortis, in order to determine the most relevant sequencing depth levels, we started from very high depth data sets for different matrices, gut, but also skin and vaginal microbiota, for example. And then by rarefying the data set, we observed the relation between the information content and the sequencing depth, and we determine the explorer, advanced, and expert levels. And of course, during the validation of the analytical process, we also conducted repeatability and reproducibility tests in order to verify the robustness of the analysis.
Thank you everybody to attend this webinar. If you have some requests, project to discuss, you have to contact our business developers and they are available for you.
Thank you and have a nice day.
And for your information, this presentation will be available for replay and we will send you the link to see the replay. Thank you very much everybody.